Welcome to the deep dive. Today, we're digging into something really fundamental, gene expression.
Absolutely central to biology.
Yeah, and a listener shared some really great notes trying to map out the key players here.
Ah,
They wanted to, connect the dots on how genes get switched on or off without getting totally bogged down in technical terms.
that's a great goal. It can seem complicated, but the core ideas are actually quite elegant.
So that's our mission today. We'll unpack the basics, focusing on things like enhancers, silencers.
Yeah.
What else was there? Insulators, reporter genes.
Promoter elements, transcription factors, TFs.
Right, TFs, and how this all leads to gene expression, sometimes even in specific tissues. Okay, let's get into it.
Sounds good. This is, a really core concept. Gene expression is basically taking the info in DNA.
The blueprint.
Exactly, the blueprint, and using it to build the molecules, mostly proteins, that actually do things in the cell.
And a key starting point, based on these notes, seems to be the promoter.
Definitely. Think of the promoter as the start here sign for a gene on the DNA strand.
Right.
It's where the main enzyme, RNA polymerase, needs to latch on to start reading the gene.
Right, but the notes mentioned this starting line isn't always, open for business. It's often packed up.
That's a perfect way to put it. Yeah, it's usually tightly wound around proteins called histones.
Histones, okay.
Yeah.
Forming those nucleosome things.
Exactly. Nucleosomes. Imagine roadblocks packed really tightly on that starting line.
So the polymerase just can't get in.
Pretty much. If RNA polymerase can't physically access the promoter, the genes simply can't be transcribed. No transcription, no gene expression.
And this packed state, that's heterochromatin.
That's the term, yes. Heterochromatin is the dense, inaccessible state.
So first job is clearing that promoter, getting rid of the roadblocks.
Precisely. That's called promoter clearance. You have to loosen things up.
Make it accessible.
Right. Transitioning from that tight heterochromatin to a more open state, which we call euchromatin, that's essential to even start the process.
Okay. So unlock the promoter. And this is where enhancers apparently come in, which sounds really cool.
And answers. Yes.
The notes say they're regulatory DNA sequences,
Big
but,
throttle.
and there's the wild part, they can be super far away from the gene they control. How does that work?
It is pretty amazing, isn't it? It seems counterintuitive.
Yeah.
But it comes down to the 3D structure inside the nucleus. DNA isn't just a long, straight line in the cell.
It folds up.
It bends, it loops, it's incredibly dynamic. So an enhancer sequence that's thousands of base pairs away linearly can actually be brought physically very close to the promoter through this looping and folding.
Wow. Like folding a piece of string to make two distant points touch.
Exactly like that. DNA origami, as you said before we started recording, so spatially they become neighbors.
Okay, that makes sense. DNA origami inside the cell. So the enhancer gets close, but what does it do then? It recruits things?
Yeah.
Histone modifiers.
The enhancer acts like a docking station. It attracts specific protein. And once those proteins bind, they can then recruit other helpers. Two main types. Histone modifiers.
The chemical taggers.
You got it. Enzymes that add or remove chemical tags like acetylation or phosphorylation onto the histone proteins.
Okay, and the other type?
Nucleosome remodelers. These are like little molecular machines that can physically slide or push the nucleosomes around, shove those roadblocks out of the way.
Okay, so the modifiers loosen the histone's grip with chemicals.
Right, changing the electrical charge, making them less sticky to the DNA.
And the remodelers physically move them.
Exactly. Both work together to open up that promoter region.
So the enhancer basically calls in a specialized crew.
A demolition and renovation crew.
Yeah, with chemical tools and little crowbars to pry open the promoter area.
That's a fantastic analogy. It works. By loosening that grip, reducing the attraction, the promoter goes from that tight heterochromatin
To the open euchromatin?
Precisely. And now the promoter is accessible. RNA polymerase can bind, and transcription can finally begin.
So the enhancer enhances transcription by clearing that path. Makes total sense.
And this whole mechanism, this ability to act from a distance, is fundamental to why different cells, say a muscle cell versus a brain cell, can function so differently even though they have the same DNA. Different enhancers are active in different cells.
Controlling which genes get switched on where.
Got it. Okay, so enhancers switch things on. The notes also mention silencers. Opposite job, I assume.
Exactly the opposite. Silencers are also regulatory DNA sequences, often work via similar looping mechanisms.
But they inhibit. They shut things down.
They inhibit gene transcription, yes. The notes got that right. They do the reverse of enhancers.
So how do they manage that if enhancers loosen chromatin?
Silencers promote the compaction of chromatin. They recruit a different set of proteins.
A different crew.
A crew that often leads to histone modifications that favor tighter packing. Deacetylation, for example, can make histones bind DNA more strongly.
So they basically reverse the process, turn that open euchromatin back into the tightly packed, inaccessible heterochromatin.
Effectively padlocking the promoter again.
You could say that, making it much harder or impossible for RNA polymerase to bind gene silenced.
Okay. Enhancers open the door, silencers slam it shut, acting from afar via DNA bending. Cool.
Aw.
Now, the listener's notes had a few other terms too. Insulators, reporter genes, transcription factors, tissue-specific modules. They didn't have much detail, but how do these fit in?
Right, those are definitely part of the bigger regulatory picture. Think of insulators as boundary elements.
Or fences.
Exactly like fences. They're DNA sequences that can block an enhancer from activating the wrong promoter or stop heterochromatin from spreading where it shouldn't. They ensure specificity.
Makes sense. Keep the enhancers focused on their targets. What about reporter genes?
Those are really useful research tools. A reporter gene is one whose activity is super easy to measure, like maybe it makes something glow green. Scientists hook these reporter genes up to regulatory sequences like enhancers or promoters. Then they can easily see, is this enhancer active in this cell type? Just by looking for the glow.
So they help us study the system. And transcription factors, TFs, we mentioned them briefly.
TFs are critical.
Yeah.
They're the proteins that actually bind to specific DNA sequences like enhancers and promoters.
The ones recruited by enhancers, or others too.
Some are recruited by enhancers, yes, but TFs are a huge family. Many line directly to the promoter region itself. They can act as activators, helping RNA polymerase bind.
Or repressors.
Or repressors, hindering it. They're the direct molecular switches, really. Enhancers and silencers often work by recruiting specific TFs.
Gotcha. And tissue-specific modules. That sounds like it ties back to the muscle cell versus brain cell idea.
Precisely. It just emphasizes that the whole regulatory landscape, which enhancers are active, which silencers, which TFs are present, is tailored for each specific cell type or tissue.
So different tissues have different modules or combinations of these regulators switched on.
Exactly. That's how you get specialization. The unique combination of active regulatory elements dictates which genes are expressed, leading to all the different cell functions in our bodies.
Wow. Okay, so it's not just enhancers and silencers, but this whole network. Insulators for boundaries, reporters for study, TFs as the direct switches, all orchestrated differently in different tissues.
It's a beautifully complex system, an orchestra, like you said earlier.
Yeah, a very intricate biological orchestra.
And what's amazing is how these relatively simple DNA sequences interacting with proteins manage this incredible level of dynamic control.
Okay, so let's recap this deep dive then. Gene expression isn't guaranteed just because a gene exists. It really hinges on whether the promoter is accessible, right?
Accessibility is paramount.
And enhancers and silencers are key controllers of that access, often acting from way down the DNA strand through looping.
Using protein crews to modify the chromatin structure.
Enhancers recruit crews to loosen things up, making transcription possible.
Promoting euchromatin.
While silencers recruit crews to tighten things down, shutting off transcription.
Promoting heterochromatin.
And we saw how insulators, reporter genes, TFs, and tissue specificity add more layers to this regulation.
Absolutely. It's all interconnected.
And thinking bigger picture, understanding this, well, it helps make sense of development, how cells differentiate, maybe even disease.
Oh, definitely. Tiny changes in these regulatory sequences. Maybe an enhancer doesn't bind its protein correctly or a silencer stops working. That can have huge effects.
Yeah, disrupting that delicate balance.
It really underscores how finely tuned the whole system is.
Absolutely. So, knowing these fundamentals gives you a lens to see biology in a, well, much richer way. So here's a thought to leave you with. Considering this really intricate dance between enhancers, silencers, and all these players, what could be the ripple effects, the really far-reaching implications, if even small disruptions happen within these regulatory parts of our DNA? Definitely something to ponder. Thanks for joining me for this deep dive.